Have you ever wondered how organisms grow? How they reproduce and even repair their skin or cells? How does this all work? That's what we'll be talking about today. In the heart of every living cells, there's a great dance happening, and this has been performed for over billions of years and still continues working until maybe the last life of living organisms. This dance is called DNA replication, and it's the very process that allows organisms to grow in a massive form, reproduce in multiple creatures, and also used to repair the damaged tissues or cells. This process also allows life to continue from one generation to the next. Let me tell you a story, but for now, let's delve at the molecular level. Imagine you were a cell preparing to divide to create two new cells. But before that can happen, it needs to make sure both cells have a complete set of instructions of DNA so they can function properly. This is where the magic of DNA replication begins. But before we proceed in the magic of DNA replication, let's have a short review about the structure of DNA. So DNA is a double helix which consists of alternating sugar and phosphate groups. This twisted ladder is formed by base pair of adenine, paired with thymine, and cytosine paired with guanine. These nucleotides hold the secret of life, the blueprint for every living thing. So that's the structure of DNA. Now we proceed to the best part of the topic, the magic of DNA replication. In the process, there are construction workers who complete the replication, and these are the enzymes, such as DNA polymerase, primase, DNA helicase, and many more will be included in the process. In order to copy, an enzyme called topoisomerase will bind to the DNA to relax the twisted ladder and to be able to continue the process. The DNA must first unwind the tightly packed coiled structure. How to do this? An enzyme called DNA helicase will enter to unzip the double strand of DNA, breaking the hydrogen bonds between base pairs, creating two separate strands, which can also be classified later as the leading and lagging strand. After that, a protein called SSB will be attached to the separate strand to prevent the hydrogen bond to reform. This separation also results in the formation of a replication fork. Replication forks serve as the actual area where replication takes place, and the two separate strands are referred to as leading strand with the direction of 5' to 3' end direction, and lagging strand with 3' to 5' end direction. To start the replication, an enzyme called primase will be attached to the one standard, which classifies the template strand. Then the primase will synthesize a small piece of RNA, copying the sequence of nucleotides on the template strand. This new synthesized RNA is called primer. After making a primer, the primase will dissociate and another enzyme called DNA polymerase will bind to the primer, and this will start the construction of the new strand of DNA. The DNA polymerase moves to add the DNA bases of the template strand bases. Adenine pairs with thymine and cytosine pairs with guanine in a direction of 5' to 3' end direction towards the replication fork, which can be classified as leading strand. At the leading strand, the replication is made continuously because the DNA polymerase moves at the same direction as the replication fork. The origin of replication in yeast is known as autonomously replicating sequences, or ARS. These include a consensus sequence, an A domain, and additional elements. As the cell enters the G1 phase of the cell cycle, the origin recognition complex, or ORC, binds to the ARS region with the help of ATP. Next, two copies of the mini-chromosome maintenance protein complex, or MCM27, attach alongside another helicase-noting protein, CDT1. This unstable complex triggers ATP hydrolysis by CDC6, leading to the release of CTC6 and CDT1. While the MCM complex encircles the double-stranded DNA, MCM now functions as the helicase. During the G1 phase, the DNA unwinds but replication does not yet begin. As the cell enters the S phase, the initiation of replication begins with the activation of two protein kinases, cyclin-dependent kinase, CDK, and DBF4-dependent kinase. DDK phosphorylates the MCM27 complex, while CDK phosphorylates two replication proteins, SLD2 and SLD3. Phosphorylated SLD2 and SLD3 binds to the DPB11 and attach to the origin complex, facilitating the binding of helicase-activating proteins, CDC45 and GINS.